And so I thought I would allude to, of course, the incredibly famous and influential in the world of modern biology series of essays that he wrote. And the first part was What is Life? The Physical Aspect of the Living Cell. And actually this was also published together. The first publication of his lectures in 1944 was also published together with another series of essays called And Mind and Matter. And so I thought I would take a bit of a liberty here and draw from the example of how Schrödinger was thinking about this incredibly interesting problem, which had so much influence on modern biology. And so many physicists who read this book, What is Life, were very much influenced to go on to really lead and create the world of modern biology. So I thought, well, I would sort of draw on those, first of all, titles by Erwin Schrödinger and say, well, what if we start to try and use the way he thought about things? So, what is life, right? Well, I'm going to have, well, what if telomere attrition causes human aging, okay? And how could mind affect this? And you're going to start thinking, well, I'm very presumptuous. And I realize this, but I'm trying to not say, oh, this is going to be in the same echelon as Schrödinger's extraordinary insights, but just to say, I'm doing this in the spirit of homage to Schrödinger. And so that's how I thought this was interesting when I started thinking about it this way. And I realized that this journey I'll tell you about did have some of the same sort of structure. So what is this journey? Well, this is a picture under a microscope of a human chromosome set, and these chromosomes are double. They're about to divide, which is what these blue sausages that look double are. Each one is a newly replicated chromosome, and the dots at the end are the telomeres, which are lit up by a fluorescent dye, which picks up a common structural component in all of the telomeres. And so the telomere, quite simply speaking, functionally speaking, is the protective end at every chromosome end. Now, a problem that might seem very different. How do we age? So we're all familiar with this time sequence, of which a few examples are shown here. And we also know that aging is a complex organismal process subject to all sorts of very interesting controls. It has great implications and so on. But the part that the research that I do, and which I'll unfold for you and many others do, is that aspect of aging, which of course is extremely unwelcome, and that's the increased susceptibility, particularly to the common and often comorbid, that is to say, occurring diseases of aging. So, for those of you who need to go to sleep, this will be where we will end up, and that's going to be this kind of conclusion here, which is chromosome end maintenance helps protect from diseases of human aging. So that's pretty bold kind of a statement here. And so I'm going to take you through how the science has evolved into us thinking that kind of thing. So let's draw from some of the central chapters of Schrödinger's book, What is Life? So we get to the guts of the book in chapter 4, and he talks about, first of all, the quantum mechanical evidence, and then there are various sections in the chapter that I've just given a few examples of here. So let's talk about, for telomeres, the basic science evidence, and then we'll talk about what are telomeres, the molecular nature, the action of telomerase, I'll tell you what that is, and the effects on cells when there isn't telomerase. So this is sort of the basic ideas. And then we'll draw from the next chapter. Again, just the structure of the chapter, which was to say, having got the basics, now what about a model, okay? In this case, it's Delbruck's model of the gene that's being discussed. And so the sections, again, are different parts of the chapter by Schrödinger, and I've just drawn some of the subheadings, the general picture of the hereditary substance, different states of matter, and comparing that with what are the facts, what is actually known. So let's take again this approach and say, well, what about telomere attrition as a cause of aging? That model discussed and tested. Okay, we're going to talk about hereditary material. Okay, hereditary telomerase and telomere mutations, as I'm going to show you, cause aging diseases. We'll see that unkept states of telomeres cause cells to malfunction in ways that predict aging diseases. And then, well, what about the facts of human life, human aging? And we'll talk about telomere shortness and how that relates to aging. So we're sort of testing the idea here. So first of all, the basic science. So let's begin at the very beginning, or if it's the telomere, the very end. And here's the telomere, and very simply speaking, the DNA at the end of the chromosomes is made of tiny repeated motifs. I'll show you an example in a moment, and it's just repeated over and over. So our chromosomes, we have literally thousands of bases of nucleotides, the building blocks of DNA, which are made up of, now I'll just home in on a few of these repeat sequences, just this very simple repeated motif. It doesn't code for proteins, it's not a gene, but what this does, and now this is just the very end part, now imagine that same kind of thing with all these T's and A's and G building blocks repeated for a total of a few thousand times, all the way at the end of every chromosome. What that does is makes a molecular scaffold, which now attracts a whole set of protective proteins that make a sheath. That's the point of these repeated sequences, is to make that protective sheath. Okay, why do we care about that? Because that is inherently an unstable protective portion of the chromosome. And the reason comes from the fundamental basic molecular biology of DNA replication, which it turns out, because of the mechanical and biochemical properties of DNA replication enzymes, they can't replicate all the way to the very end of a linear chromosomal DNA, such as the chromosomes DNA in all of our cells. And so what happens is that, left to itself, DNA replication would continually fail to replicate the very ends of the chromosomes, and the prediction, recognized by Watson and Olovnikov in the early 1970s, simply based on this DNA replication machinery biochemistry, was that, okay, every time that linear DNA replicated, it would get shorter and shorter and shorter and eventually dwindle away, and maybe you'd lose something at the end. Perhaps that would even cause senescence, as Olovnikov very presciently thought. And so there was a problem here. So the problem was solved, and I won't go into all the steps that led us to this, by using a biochemical approach. And what we did, and this is my student Carol Greider, shown at the time when she was wearing her California uniform you see here, and she was a student in my lab, and I had got a certain amount of evidence that said, well, maybe there would be something that actually adds DNA to telomeres. So the bottom line was we made a synthetic telomeric DNA, and that's little building blocks of G's and T's, which is the telomeric DNA sequence of the particular system we were using. We then put in this extract from Tetrahymena, which is an organism that lives in pond scum, hence the pond scum, and found that it could support the elongation by addition of new nucleotides to that DNA primer that we'd added in there. So then the question was, did that matter? So we turned to our pond scum system, and this system, interestingly, it can divide pretty much forever, so long as you feed it, talk to it nicely, so on. So it also had some telomerase in it. That's actually how we found the enzyme. We used this organism because it has a very large number of very small linear chromosomes in it, so we reasoned it was a good source of telomeres and maybe a good source of the enzyme, if such an enzyme existed, and that's what we found it did, that would maintain telomeres. So now, cells are immortal, plenty of telomerase. That's a correlation. Did they have anything to do with each other? The test is, of course, genetics. So by making a single mutation in one of the components of telomerase, we were able to show that now, when you crippled or inactivated that telomerase, now, as the cells kept dividing, the telomeres progressively shortened and shortened, and eventually the cells, when the telomeres got too short, stopped dividing. So we'd struck at the heart of the ability of these cells to be immortal by this very simple single change to a single chemical building block within the telomerase enzyme. And we knew what that block was. So basically, this was the bottom line. Immortal, mutate telomerase, the cells became, quote, mortal. And so I go through this historically, partly just to show you how these ideas don't come from nothing, but come from experiments, but also because that was the general principle. And so now the whole thing is the elaboration of all of this and learning a whole lot more about the system by basic science approaches, and then starting to try and think about how this plays out in humans. Okay, so we have the telomere and the repeated sequences, which, as I said, make this nice scaffold for the protective sheath. Those proteins are shown in colored blues and aquas and yellows and grays and so on. That nice sheath of proteins about which a great deal is known. But the problem, as I said, with telomeres is that they are subject to shortening. Now, when they get too short, a lot of research from mini labs doing basic science research in cells, looking at molecules, looking at cell responses, is that the bottom line is that that very short telomere now no longer is recognizable as a protected end and is now sending a warning. Molecular signals get sent from that too short telomeres, and that has a very clear set of effects. So this has now become very well studied in human cells. What it does is that warning sets off a whole state in the cell where the state of the cell becomes different. For those of you who are aficionados of this, the transcriptional program, the whole program of how all the genes get expressed is altered in that state of long, sustained warning signal coming from the uncapped, as we call it, telomere. And those cells start to do very strange and bad things. They start to secrete pro-inflammatory and pro-tumorigenic factors. So now they're like rotten apples. They can't, exist normally. They're now doing things that would affect cells around them. So in the body, if you have cells like this, they can be deleterious because of the secretion of such factors. The cells get a signal that says no longer keep dividing. There's something wrong, they put on the brakes. So their renewal capability, if their cells that normally are replicating, is stopped. Furthermore, if certain other things go wrong in the cells, the DNA at the ends, this unprotected DNA at the ends of the chromosomes, can start sticking like chewing gum to other unprotected ends, and this will make the genome very unstable. So I just want to show you what happens if you look inside a cell and say, what's happening to the telomeres when this kind of thing goes on? So I've called this jittery telomeres, so you'll see why. So we can visualize telomeres in living cells. So I've just shown you a kind of an outline picture of, this would be a cell outline here. This is very faint, the slides of it. Okay, they look beautiful on my screen, but anyway, it doesn't matter. Here's the nucleus, and here's a couple of chromosomes. And what you can do is with a fluorescent protein that just homes in at telomeres, you can light up the telomeres. So in live cells that you've put under a microscope, and so you just see the telomeres. So now, of course, there are lots of telomeres in a human cell, typically almost 100. So now here's this whole cloud of telomeres in the single nucleus of a single cell. And so what happens? Now I'm going to point you, see this, this arrowhead here. Just have a look at this group here, first of all. So I'm going to now play the movie. Now you get a pretty general sense that a lot of movement is going on, but I actually want to point out this one here. Look at this one where the yellow arrow is pointing here. This one here. It's very steady. It's steady as a rock. And so we found telomeres were in two classes, steady as a rock and jittering around. So we analyzed the molecular components that were responsible for these behaviors, and essentially we found that when the telomere is getting too short, it starts behaving in this very sort of jittery fashion. It's quaking in its boots, right? So this is an interesting sort of reality. This is what happens in the cells. And biophysically, we don't understand all the details of what is going on, although we understand some. But we also don't really understand, does this have a purpose? But we can certainly see a jittery telomere from a stable, well-capped telomere, and the well-capped telomere is rather still and quiet. So now, imagine inside our cells. Is this going on in humans as we age? Think of our cells, imagine, do we have telomeres that are jittering like that in some of our cells? So it's fairly clear that as we age, that this kind of thing does go on. So now we're going to turn to, well, what is known actually in human cells? So we'll turn to this question of, okay, so I've given you some very basic ideas of telomeres. They're very normally stable, but if you uncap them, if they get too short, they become unstable, they get jittery, they make cells malfunction. This is all in the lab, this is all good. What about in humans? So the first thing is that a lot of studies show that if you look at human cells, in normal adults, let's say, the ones who will be aging, the telomerase is often rather limiting. And in fact, there is a general trend to telomeres shortening with age, and I'll show you the actual pieces of data about that. But basically, they do indeed show evidence that this progression goes on. And as I said, this will lead to consequences. What's called senescence, that's when the cells are ceasing to divide. Remember I said they will no longer be renewing. The malfunctioning, they've changed the function, the program is now different, they start misbehaving. I told you they secrete pro-inflammatory factors. They even start secreting, they start misbehaving in the pancreas. A cell like this that's got a very short telomere signaling has been shown to not be able to respond properly to insulin signaling, for example. And then, as I said, a risk of genomic instability. So here is this progression that you can see evidence of happens in humans. Particularly, you find evidence of such cells as people are getting older. So the question is this really accounting for what goes on in the trajectory of human life? You know, does the candle, so to speak, burn down by analogy with this telomere wearing down as cells are dividing in humans? And, you know, eventually does it burn down to the point where you die? An attractive model, because there is a sort of built-in delay to this process. Okay, so we're very hypothetical. And we also have to think about, well, what can save the cells? So remember, I've told you about the wearing down, but actually there is telomerase in cells, and you can measure it. And it is, as I said, often low, but it is active in certain stem cell populations, also in the cells that give rise to the germ cells that will give rise to the next generation. Makes good sense, because you don't want telomeres shortening from generation to generation normally. But it's also even present in, once you look hard enough, you can find it in a lot of cells that are not stem cells. So here we have a situation where it's not just on or off, like I showed you in the tetrahymena experimental situation, but much, much more nuanced. And the more you look at the telomerase enzyme, which I've now depicted as this sort of little creature at the end. So this is a chromosome, this is the DNA, and this is the very end of the DNA. And here there's the telomerase working, and it's a lovely enzyme because it does something very unusual. It uses a small template in what's called an RNA molecule, and the nucleotide bases in the RNA is shown here. And it actually uses them as a template for copying by Watson-Crick base pairing rules, copying in nucleotides that get added to the DNA end. It's a very unusual, interesting mechanism. And so these are just two of the core components, the RNA core that has the template and the core protein, which is called HTERT for human TERT. And this is HTR for human telomerase RNA. But it's true across most eukaryotes. It's very common to have telomeres maintained by this quite conserved mechanism. Now, the more you look at the control of the telomerase, I'm just going to add this very complicated statement here, and that is that the more you look at the control in human cells, you find it's very nuanced. It's under almost every sort of control molecular mechanism you could think of. Transcriptional, just for the specialist here, splicing, which is how the transcribed information from the genes get further rearranged, assembly of the protein and the RNA and other components I haven't shown you, trafficking, how it moves between different cell compartments, signals that regulate many processes in cells are also heavily impinging on telomerase. It's one of the most highly regulated things you can think of. It really looks as though humans are keeping it under some tight controls. And so it's very hard to make an a priori prediction about what's going to happen when you've got so many different complex controls going on. But we can set ourselves a kind of framework for this, and we can say, well, what will happen if you have, like a tetrahymena cell that's immortal, plenty of telomerase? Now, the addition by telomerase can be then balanced by the shortening processes. So if you have addition, it'll keep adding DNA, and the shortening, which is part of this DNA replication problem, and other things also cause shortening too, that if they're in balance, then pretty much overall the telomere length will be maintained and the cells will just keep on dividing because the telomeres won't run down. Stem cells have this kind of general feature, although not always. And even if you look in some of the stem cells in adult humans, you find actually it's not always completely in balance like this. But very seriously, human cancers have often upregulated their telomerase enough that they just keep on dividing because their telomeres never run down. So that's a sort of dark downside of having enough telomerase, is the cells you don't want to have replicating, which in this case are tumor cells or cancer cells, and which have a great many other things wrong with them as well. But one of the things they very commonly have in humans is that they have plenty of telomerase, in fact, more than the normal amount. And that is one thing that's making them have their pathological properties, because they just won't go away, they keep multiplying. But you can imagine other ways in which the balance can be set up the other way, and that actually happens in our immune system, in certain cells of the immune system, where the balance actually, at certain stages, the telomeres actually get longer in normal adult cells, in particular stages of development of certain immune cells. So the balance can go that way. But more commonly is the situation here, where you can actually see that the telomeres over time will get shorter. You know, if you have some telomerase, though, then the telomeres may take a while to get shorter and shorter, because it's constantly been sort of, you know, counteracted by some telomerase, but eventually they'll get to senescence. However, if you had less telomerase, and if all other things were equal, that's not, of course, going to always be the case, but we're just setting it out here, then you might expect telomeres to get shorter and shorter faster. And that, of course, is going to be under two very general kinds of controls. I've told you it's a very elaborately controlled process in cells, but we can start to think of two kinds of influences, and which will be very informative in this question of, does telomere attrition have a role in aging and in the diseases of aging? We can think of genetic, and we can think of non-genetic, such as environmental or life factor influences. Okay, so now we can sort of set up the problem here. So, to do science, you have to be able to quantify, and so you have to be able to measure telomeres. So this is now quite well developed, and there are various ways of doing it very elaborately in the lab, where you can look at every telomere and every cell if you want to. But in practice, if you want to look in humans, which is now what we're going to turn almost exclusively to, if you want to look at telomeres in humans and do studies in clinical kinds of cohorts and studies, you need a practical method for just measuring telomeres in realistic samples with realistic costs. And so the measures that work quite well are just to use molecular ways of doing this, where you measure the average length of telomeres in the sample or the population of cells, let's say from an individual person. So you can measure telomere length, is the bottom line, and the methodologies are quite good. So all the things I'll be talking about is where you're simply using a mean measure of telomere length. But you can imagine in this progression of telomeres getting shorter and shorter, that the mean telomere length will capture some parameter about that population of telomere lengths. And indeed, it's turned out, as you'll see, to have quite a lot of interesting predictive value. So let's see what happens if you actually look at, say, a very typical study. Lots of studies have been done like this, where you take a few hundred people and you measure the average length of the telomeres in white blood cells. So you get two things. One is a general trend down. If you look at the age of the person whose blood sample you're looking at, you see that there's this trend downwards. Okay. However, you can also see, and this is the point, that there's a great deal of scatter within any one age. Chronological age accounts for 10% or less of the variation in white blood cell telomere length. So you might sort of throw your hands up and say it's hopeless, right? But you can also try and ask, well, what factors are accounting for this variation between individuals? Let's say all the people who are, 40 years old, there's a big range of telomere lengths, even though this trend downwards in these sorts of typical studies of a few hundred people is really always, very, very, is robustly seen. But what's the significance of all this telomere length data you get with people? Can we make any sense of it? What accounts for it? So I'll tell you one kind of study where you can start to bring a lot of information together by, if you've got enough samples. And so this was, we joined in a study which is called the Kaiser Research Program in Genes, Environment and Health, and they were looking at the genomes of 100,000 people and looking at genetic variability of all of these 100,000 people, and also tying that in with their electronic medical health records, of which they had, most of them were at least 20 years old, records going back continuously, for 20 years for about half of these individuals at least. The individuals ranged from about 20 to 95 years of age. There was also a lot of information about where they lived, their environment, their life, and so on. So there's a lot of data being collected. And we joined in and said, let's measure telomere lengths in the DNA in these 100,000 people so it can get related to all of this very rich source of data. Now, this is not the only such study being done. There's certainly studies in cohorts in the UK being done of large numbers of people. This may be one of the larger ones. But this is the kind of thing that gives you the kind of statistical power to kind of stand back and look, from 40,000 feet up and just sort of look at a swath of humanity. This swath of humanity happened to be in the western part of the United States, but very diverse group of people. So let's look at age. And so this was interesting because for the first time, and I'm just showing now the means without showing you that scatter, but the means and the standard errors. So it's really quite nice and accurate because we have at least 500 points for each of these ages here. We're looking at telomere length on the y-axis and age of the donor, the person who is participating in the study, on the x-axis. And what you see is that expected decline, but then we saw something that nobody had the power statistically because they didn't have the numbers to see before, and that is that once you get to about 75 years of age or something, then actually they're longer and longer and longer and longer, okay? Now, most mortality in this population is happening around the 75-ish years old. So this is now reflecting more and more likelihood of survival or some sort of selection or whatever you'd like to call it, right? So no one had seen this uptick before. Another interesting thing was we were just looking at the 100,000 people, just trying to see what's been seen before and what we could see new. If you divided people up into men and women, interestingly, there's a big split right about postmenopausally between women and men. Both show, again, this increase in average telomere length in these cross-sectional. These are just one time point per person. The longer the person lives, the more likely they are to have longer telomeres. So just looking at these large numbers, you can get this sort of picture from 40,000 feet up. And then, of course, you can start dividing it up in all sorts of ways. And I wanted to show you a couple of points. So what about smoking? Well, if you look at all these 100,000 people and ask about their smoking habits, what you see is a very clear dose dependency where you're looking at the telomere length and you can see it's lower and lower the more the person has smoked in terms of the calculated pack years, their smoking history. And I can't resist juxtaposing that finding with, and this is repeating findings that have been reported elsewhere, so I'm going to juxtapose this with a rather recent study where people were looking at identical twins who smoked less or smoked more. This just came out as a recent study. Here's the twins, two members of two twin pairs who smoked less over here, and the twin who smoked more. It's a bit hard to see in this light. I'm sorry, it's a bit dim here, but basically these people looked a lot older, right? So now this is a different study, but it's very tempting to sort of juxtapose these two and say, look, we've got equal genes here, but the smoking is, you can see these people visibly older than the twin who's genetically identical but who smoked less, and there's this real dose-dependent response for telomere length. Another interesting study has related to something you might think is completely different. This seems like another world, but I think is going to turn out to be very important to think about. And that is a study where it had been shown by Andrew Steptoe and colleagues in the UK Whitehall civil servants cohort. This is Michael Mahmoud's cohort. They've been studied for many years. And if you look at telomere length in these individuals, what you find is that the more education a person has had, so on the left, they didn't finish high school, what we call high school in the United States, secondary school. If they did their O-levels, they did their A-levels, or they went to some sort of university or college or tertiary education. There was this graded response. And even if you adjusted for things that you might expect to be a result or a consequence of not having finished your secondary education, such as their employment and their health behaviors, their income, and all these triple bars are just showing you that nothing shifted. So this was very robust and didn't depend on things and get explained away by things like, a better job with a higher household income or better health behaviors or something. So we wanted to look at this with the 100,000 people, and amazingly, it just replicates really well. These are the people who didn't finish high school. Again, we see the graded response. So this has been seen in other studies as well. And so the point is that there's something pretty, international about this kind of pattern, and we were seeing it with the 100,000 people. So does this matter? So hence the end-to-end title here. Well, does this actually have any relationship to death? Well, you could ask that with the 100,000 people, because what happened was that the DNA was actually collected back in 2009. So the clock ticked, so the DNA length was measured from DNA samples that were taken from the person here, 2009, then the clock ticked and 3 years went by. And you could say, well, who died? Now, this is the whole cohort, so they range from 20 to 95 and more. So, it includes a lot of young adults as well. So about 2,500 people died. And then you could say, how did those people who died relate to what their telomere length was like 3 years ago? Compare it to the mean baseline telomere length. So baseline is at the beginning here. So what you saw was quite interesting. The people who had telomeres that fell into the bottom quartile of the length distribution, their probability of being dead is set to 1 here. And so what you can see is that people who had longer telomeres, they had a less chance of dying. And then you could say, well, maybe it's just because they smoked differently, or their age was different, or it was their race or their education. I showed you those are important. I didn't show you race, ethnicity. That plays a bit of a role too. Or their cigarettes, So then you could correct for all of that. And that is the orange bars. And you can see it made no difference whatever. So this is rather independently predicting, even in 3 years. Now, I want to just put the numbers into a bit of context here. So I'm going to show you the same data set, but now I've plotted it a very different way. It's plotted, now you're actually looking, here is time. You're going out to about 3 years from left to right. Telomere lengths is measured. And then you're looking cumulatively at the people who with different telomere lengths at the beginning, with what frequency they died. And so what you can see is that the chance of somebody surviving was much this is actually a negative axis. And so the more chance you had of dying, the more steeply the curve rose. And so the bottom decile really pulled away, actually, from the other deciles. It's the same data set, but I've just shown it now. So it's the bottom decile, the people in the bottom 10th of telomere length. Now, the scale is a little hard to see here, but basically, remember, only 2,500 people died out of 100,000, because this was, from 20 to 95. So most people, of course, are not going to die in 3 years. But I just want to juxtapose this and show a very similar kind of thing was found. This is Honig et al. Study with now much older people who started off at 66 to 101 in age when this study. And now if you look at telomere length at the beginning, and they followed it out now for 15, 16 years, and they see this very nice graded relationship with the longer your telomeres are, the less likely you are to die. Now, I'm showing this data set too, because now I want to show that this is a pretty big difference. So the proportion of people who died, now if you look, say, at the 14-year time point, it's now, if you're in the longest quartile, only half had died, but if you're in the shortest quartile, 3/4 had died. So now we're sort of talking about fairly big numbers here. So obviously, it's not the only factor that's going to be affecting whether you die, but what we're seeing then is a statistical relationship here. And I think with this 100,000, we can get a certain good set of sort of confidence that this is statistically predicting future mortality. It's not a crystal ball, but it is statistically telling you probability of dying. And you could even start discerning that after 3 years. Okay, so dying, what do people die of? Elderly populations, by and large, tend to die of, the major common diseases of aging, the comorbid diseases, which are very commonly found in populations, and particularly in the aging demographic. And what is found is a quantitative relationship between telomere shortness and diseases, getting these diseases. I'll elaborate a bit more, but they include the very common sort of maladies, syndromes, and diseases of aging, impaired immune function, certain cancers. I'll talk about cancers a little bit more in a specific way in a moment. Lung disease, heart disease, diabetes, vascular dementia, osteoarthritis, risk factors. All of these quantitatively are related in terms of their risk or their incidence, depending on the study, with shorter telomeres. Okay, but that's a double-headed arrow here. It doesn't say anything about causality. And as I just showed you, mortality risk is predicted by shorter telomeres. I just showed you that. So now we want to consider causality. Okay. So when we think about these diseases, which account for and cause a lot of mortality in elderly populations, we know there's going to be a lot of inputs. There will be non-genetic inputs, and I've listed a few categories, environmental, life events, behavior, so on. And then there'll be genetic inputs. And then there'll be interplays between the two. Genetics is particularly helpful because by patterns of inheritance, genetics is very informative about causality. So we've learned some genetics lessons about telomeres and their maintenance and diseases. And so I'm just going to summarize. This is a lot of people's studies here, but they've really collapsed into a few lessons. So the first and the simplest one is the situation if people have rare mutations, fortunately rare mutations, but they cause you essentially to have only half the amount of telomerase level. It's done through different molecular mechanisms. Sometimes it's different genes in telomerase or in telomere, protein protective genes that allow telomerase to work. But the point is, you get a simple situation where you have about half the amount of telomerase. So, the roulette wheel of genetics has spun. This person, unfortunately, got the bad luck to have a gene that is now inherited and is causing about half of the amount of telomerase. And there the causality is very clear. They have much shorter telomeres than their age group, really noticeably shorter than their comparable age group. And there's a very clear disease impact. And it's a set of diseases and a set of syndromes which are summarized here. Now, they die early. So we saw, in the population, earlier mortality is correlated with telomere shortness, although there was no causality implied in that. But we see immune system failure, certain cancers, pulmonary fibrosis, cirrhosis, and even it's now emerging in the clinical studies, these people are being susceptible to diabetes at unusually early ages. So what we're seeing is a real overlap with a set of the common diseases. Now, their onset is different and much earlier. And in fact, individuals don't live a full lifespan usually. So basically, the causality is very clear. We know that having not enough telomere maintenance can cause these kinds of things. So that's extreme, though. So what happens in the population at large? And a wonderful study coming out of the UK with Spector and many colleagues, Codd et al.'s paper this year, showed a very interesting thing. When you look at populations of people, now there's a lot known about telomere genes and telomerase genes. So if you go in and look at those genes, we know that all genes have common variabilities from person to person. And the common variations are called SNPs for short, doesn't matter why. The point is that the common variations in genes can be seen, and you can look for patterns. And so what these authors did, Codd et al. Did, was they said, well, of the genes that we know from all the molecular and cell studies are involved in telomere maintenance very directly, telomerase or parts of telomeres, are there combinations of SNPs that have an impact on whether the person has a risk for diseases? And remarkably, they found that that was the case. So we know these genes are affecting telomere maintenance. They have an impact, and particularly the ones they pulled out were pulmonary fibrosis and coronary artery disease, common particularly in the elderly. So if you believe genes are causal, well, this is starting to look like a kind of causality here. There's a contributory aspect to this. Another interesting kind of study, but going in the other direction, is the kind of thing where if you look at people who live exceptionally long, and there's actually known to be a lot of genetic component to this, but what are the genes that count? So people have been looking at groups of genes and asking in different kinds of functions, what groups of genes have biased common variations, SNPs, in centenarians compared with the population at large. And interestingly, these are telomere and telomerase related genes again. Now, presumably these are ones that are giving you better maintenance of telomeres. But what you see again is this kind of correlation. So it's not all that strong, but very suggestive taken together with what I've said before. I want to spend a moment to talk about cancers, because cancers have presented what seemed to be a real sort of complicated picture in terms of telomere maintenance and what's going on. And it's been hard, but it's starting to make some sense now. Now, I mentioned to you that the people who have not enough telomerase, because of those rare genetic mutations, that they are subject to some cancers. And in fact, these people who have just 2 times too little telomerase, remember I said they have half the amount of telomerase, that's it, just half, which is still quite a lot, right? They are very subject to getting certain subclasses of cancers, like 1,000-fold risk for certain hematological or squamous cell or gastrointestinal cancers. It looks like cancers in cells that have to replicate a lot from stem cell populations that then run out of telomeres and get genomically unstable. That's the sort of logic that people have for these, and that's borne out in mouse model studies of this. But what's also emerged from a set of papers that just came out this year is that I've just summarized all of this information. If you have mutations in the DNA that make you have just a little bit too much of that core telomerase protein, the one that's called H-TERT, just nudge it up. It's not a mutant protein, it's the same protein, but the mutation nudges up the amount by just little as 2-fold. That is a very strong promoter of melanomas and brain, liver, and bladder cancers. And you find germline mutations, rare mutations in one family studied in which everybody with this mutation that just nudged up telomerase got melanoma. But then if you look at cancer cells, which have thousands of mutations, the most prevalent class of mutations were these mutations in melanomas and brain cancers, and then pretty prevalent in liver and bladder. Not in prostate or breast, by the way, so that's not every cancer. So now you can see we're sort of stuck in a difficult place, we humans, because, okay, too little, so we're sort of, balanced here. This is not a good place to be messing around with telomerase, is the message I get from this. Especially small effects, could accumulate over years and years and, maybe tip the balance one way or the other. So that seems to be what's going on with the cancers. But if we step back from all of what I've been saying, the genetic mutations, though, overall have been saying too short telomeres do have an impact on disease. I mean, apart from that complication of those sorts of cancers I've told you with these telomerase mutations, that this seems to be a sort of general conclusion here. So now, what about in the population? So now let's move from genetics and just say, well, let's look at people. As I said, telomere shortness has been associated quantitatively with many diseases, but let's pose the question in a different way. Let's say if telomeres were in some way contributing to disease progression or onset or causing disease in some way, you might expect the telomere shortness to occur before the disease appeared or had its onset, right? And in fact, that's found, and I'll show you some of the data for it, but just, some cancers you see it for, cardiovascular disease and dementias and diabetes. So that's a bit abstract. Let me just show you a couple of studies that really make this point. And as I said, mortality, of course, also is predicted by telomeres being shorter, even some years earlier. So here's a study. This was Willite et al., and they're looking at about several hundred people. And so telomere length is measured, and this is time axis on the x-axis. And they're divided into different telomere categories, the shortest or the middle or the longest telomere, so divided into thirds. And what you see is the people who have the longest telomeres are much less likely to either get a diagnosis of cancer in that subsequent, it's 10 years out here, or die of cancer. It's about 11 times less likely to die of cancer in these 10 years than the people who had the shortest telomeres. So it's this graded response. Now, the same cohort also was studied for cardiovascular disease. And again, it's divided into different categories here: cardiovascular disease overall, strokes, myocardial infarctions, and vascular death. And again, you see the same graded prediction, telomere length measured at the beginning, and then 10 years go by, and who got a diagnosis of one of these things, myocardial infarction, or died of a vascular disease death. Furthermore, this Honig et al. Study looked at dementia incidence. And again, telomere length is measured at the beginning. This is now in years, so it's out to about 12 years here. And the people with the longest telomeres were less likely than those with shorter telomeres to get a diagnosis of dementias. I won't show you the data for diabetes, but we've just recently seen that in a study of relatively young adults, 5 years before. If you had telomeres that are shorter, you had actually over 60% chance of getting diabetes within the next 5 years in this particular cohort. So we're seeing that telomere shortness is predicting future incidence or diagnosis of disease or mortality from the cancers, and also cardiovascular disease, dementias, diabetes. So what does this do for our causality argument? Well, you know, we want to be very circumspect here. I mean, it's occurring before the incidence or diagnosis of the common disease of aging. So I would put that in the category of, well, you know, it's consistent with a contributory or causal role. It's certainly not proof by any means, but at least it would be one requirement. If telomere shortness was causing disease processes, it would be reasonable to expect it to be seen before the disease actually makes itself manifest. Okay, so overall genetics has told us this causality here. So we do have that sort of general picture that's starting to emerge. So that's genetics, and as I said, things in terms of disease development will consist of genetic and non-genetic components. So now let's turn to the non-genetic components. You know, it's all very well to talk about genetics, and I'm sure if we could choose our parents well, we would have some control, but, you know, so what about things that we can do something about? So a very interesting area has emerged, and that's the area of chronic, and I mean severe chronic psychological stress. And I'll give some of the situations in a moment. Now, this has a long and rich history of being related to the likelihood of people developing certain common diseases and heart diseases, one particularly well-documented example. So several years ago, my colleague Elissa Eppel came by and said, what happens to telomeres with people under stress? And we said, you know, nobody knows. And so this started a whole series of wonderful collaborations that we've done. This is Jolyn in my lab, who has led these studies from the telomere maintenance viewpoint. And many other groups have been doing research in the same kind of thing. So I'm going to summarize work from a lot of different groups here. But the thing that really got me into this was that at the very beginning, when we first did this study, we were looking at a situation that sort of couldn't help but, you know, move you, because this was parents, mothers in particular, of a chronically ill child, her child that she's the primary caregiver for, and often in the US you don't get much help in this situation. And we found that the people in this study, which were both caregivers and control mothers, they had less telomerase if they were under high stress, which is the red bars, and less telomerase and shorter telomeres than those in the low stress group. And, you know, this was corrected for various potential confounds. And furthermore, the number of years they'd been in that situation, which is plotted here, the number of years she'd been in this objective stressor situation, you know, had a quantitative relationship to telomere shortness. Okay. So that's good, but that's one study, and, you know, it could be statistical accidents, right? A lot of scatter. So then we said, well, the important thing is that you replicate. So we then chose another situation. So Elissa Eppel designed this study with women caregivers who are now older people, and they have a family member, often a spouse with dementia. So, and again, she's a primary caregiver, and then there was control group too. So we looked at a lot of things that would sort of look very soft to you, like the perceived stress, where the more you go to the right on the x-axis, the worse the situation or the distressed behavior or situation is. Pessimism, for example, eating ordered severity. And we looked at those as people on these scales, often self-report. So you think very soft. And we looked at telomere length in these individuals, all blinded, of course, as these studies do, and found these relationships. So it was really very consistent that telomere shortness was correlating with the worse measure, worse meaning, you know, the most sort of stress-related kind of measure on these various kinds of scales. And then these studies have just, you know, multiplied and multiplied in different groups. And so I'm listing some of them here. So I've told you about perceived stress, caregiver stress of either the mothers or the dementia caregivers. Also, it's emerging major depression is relating to telomere shortness, particularly the lifetime duration of it is particularly related. Former domestic abuse, even if a woman has been out at least a year from the situation, the degree of telomere shortness we measured in her was proportional to the length of period she'd been in the abusive situation. And then, and I think this is starting to be very interesting, cumulative exposure to childhood traumas, even though you're looking in adults here, but these are childhood traumas, very severe traumas that happened in childhood. The more of them there were, the shorter the telomeres in adults. And interestingly, as I showed you, also less education. So we talked about less education before. So now, cumulative exposure to childhood traumas. So we're looking at adults, way later. So this was sort of earlier than, the end result of less education. But you can go even earlier. So now we're finding that intrauterine maternal stress exposure, it's the baby inside the mother's uterus. If the mother is undergoing severe stresses, which were documented stresses, that's related to her newborn having telomeres shorter right from the get-go, and the telomere shortness persisted into young adulthood. That's as far as the studies have gone so far. So what we're seeing is these non-genetic influences can be related quantitatively, and this is quantitative relationships to these kinds of parameters, to things happening really early, pre-birth even, with we get these persistent effects, which persist into late adult life. So the question, to what degree is this going to then account for risks for some of these common diseases that we showed are related to telomere shortness? So we have to start breaking the question down. So there's really good evidence that chronic stress, I think we can put an arrowhead here. This looks pretty good. I've also shown you some good, at least genetic evidence and supportive, consistent evidence that telomere shortness is also related to disease impact. But now we want to connect them all and say, how might you connect these? What is quantitatively important? Does the chronic stress have its impact purely through telomere maintenance? I'd be very surprised. I don't think things are ever that simple. Other pathways, And then if you have a disease situation going on, is that now feeding back on the telomeres too? So all of these are important to start dissecting out and trying to understand. And particularly, it's important to understand, well, what are the sort of quantitative rate-limiting steps? If one of these arrows was very minor, you'd say, well, okay, it can go on, but maybe that's not the thing we should pay attention to. So just to sort of satisfy you that you can really dive into these questions at a cell molecular level, in fact, there's quite a lot of information from cell biology, from studies in organisms, that let you start to draw pathways. So the brain is clearly where the stress perception will begin, and there's clear evidence this can dysregulate your cortisol, a stress hormone. There's good biochemical evidence that we and others have that will actually lower your telomerase in normal cells. It'll affect complicated pathways like, P53, BRCA1. I'm not going to go into the detail, but just to show you that there's plausible molecular pathways by which this effect in the brain can be seen to act on cells in ways that would lead to shorter telomeres. That, in turn, if the telomeres start running down and the immune system can't maintain itself, then that will lead to immunosenescence, which is a fancy way of saying your immune system can start getting exhausted. It starts getting very pro-inflammatory at that point. That increases pro-inflammatory cytokines, oxidative stress. That, in turn, we know in the lab, in the test tube, can lower telomerase activity. This will also reduce natural killer cell activity. These sorts of things are all things that are predicted to have an impact on some of these disease processes that I've been outlining. So this is not just abstract. There's a lot of concrete kinds of bits of evidence here. But I reiterate, it's all very well to have all these mechanisms, but you really need to understand which ones are those that are the rate-limiting ones, and therefore to focus on. Because what we want to know then is, can we affect this balance? So the observations are that we do know that there are a number of things that we can say pretty likely are causally making telomere maintenance worse. And I've outlined a few of those for you already, like smoking and severe chronic stress and childhood trauma. Now, there's also observational studies, and that's mostly what they are, which also relate better telomere maintenance to certain things that now are, there's some influence that can be exerted over here. Education, I haven't shown you the data for exercise. There's a lot of data that relate exercise to better telomere maintenance. Interestingly, omega-3, both observationally and in certain very early studies of placebo-controlled randomized prospective trials, you can start to see omega-3 levels are having a salutary effect, and stress reduction is starting to emerge. Now, most of these studies are actually observational, and the gold standard is to have placebo-controlled randomized, prospective trials. Those are in their infancy, but these are giving us very good hints about at least starting points. And small pilots have been done, which are quite provocative because they do indicate that there's some effect possible, but they're all small and quite pilot right now. But the observational studies are really quite robust. So let's just finish and step back from all of this and say, well, why would we care about this? So let's think of the aging demographic of the world, of individual countries and certainly the world, and what's happening in these major diseases, which are still such a challenge, these chronic diseases of aging, is that they go through stages from pre-disease to advanced. And what we do typically is we wait for them to become advanced, and then we treat them. So it's like the firemen rush heroically and with great skill and energy, put the fire out. But that's not the ideal situation, and often the house can be damaged. So what's happening is that a lot is being learned about the kind of things that is going from the pre-disease to the advanced stages, be it in cancer, be it in the development of things like cardiovascular disease. The stages are being more and more understood. And so now it's more and more reasonable to start saying, well, let's say as we work backwards to earlier and earlier stages in the disease of understanding what's going on, then what we'd like to do is to preempt the disease, if possible, right? Leave the house, if you will, intact. And so that's where I think that this telomere analysis is starting to fit into this, because it's one kind of factor we can isolate. So what we're seeing is obviously risk for age-related diseases and mortality obviously have a great many inputs. And I've listed some of the aging-related diseases that are particularly problematic in terms of adding to the burden, both personally, societally, family-wise, and so on, of aging. And what we've seen is that telomere attrition is certainly related to these. In some cases, we can make a case that it's contributing to these. It's clearly not the only contribution. We have genetic inputs, we have non-genetic, and we have all the interactions among them. So that's the kind of picture where I think I'm putting the telomere attrition in the middle because it's got a kind of commonality, because it's right across the body that it's working. And then it's playing out in different kinds of organs and different diseases, but it's really a sort of common underlying idea. So the idea is that this is stress-related. We think it's malleable. We see a lot of observational evidence that it's malleable, but we want to understand better how quantitatively malleable it can be in terms of real interventions. And we think it is contributing to age-related diseases. So let me just summarize the take-home. So compromised telomere maintenance, the basic science says it impairs replenishment and function of human cells. And so there's a lot of clear data about what's going wrong with the cells. And then we think it's adding up to something that I'd like to think of as a root factor. It's underlying, and it can certainly very likely interact with the mechanisms of other inputs that cause the main big burden diseases, the long-term comorbid diseases, the chronic diseases of aging. And then fascinatingly, the brain is having a big input into this that we can see in these quantitative effects as read out by chronic psychological stress, severe, prolonged chronic psychological stress. And those include the sorts of psychostress, social stresses that I gave some examples of. Interestingly, though, it does include maternal stress. So, I think there's very interesting sort of even health and policy-related issues to think about, all the stages of life here. This is not something just in aging, that we need to think about all the stages of life. And we think that it's malleable as well. So I will finish, and I want to thank you very much again for inviting me to be the Schrödinger lecturer this year, and thank you for your attention.
I think your microphone will enable you to speak. Thank you, Elizabeth. Elizabeth very kindly said she will answer some questions, and I'm sure after a lecture like that there'll be many, many thoughts and comments and questions people wish to ask. We don't have an enormous amount of time, so the most welcome sort of questions will those that be rather concise and succinct. The less welcome sort will be the very long questions or very long commentaries that don't really go anywhere. So just with that thought in mind...
the same goes for answers as well.
No, long answers are always the best. That's always the case. There are, I hope, some roving mics. Have we got roving mics? Yes, we do have roving mics in green T-shirts. So I will try and conduct affairs appropriately. Let's have some hands up. It'd be quite nice if you said who you are and, again, those short, concise, well-thought-through questions. So here we go. Hands up. One at the front here. Hope it works.
I'm a mathematician, so I know very little about biology.
Just say who you are.
My name is Dr. Julia. It's an extremely interesting talk, but I couldn't stop thinking about how does it relate to heart rate? Because usually if you're under stress, the heart rate increases. Does that mean that the cell turnover increases? How does it, what happens?
Well, the heart rate increases because of inputs from, curiously enough, the vagal nerve coming from the brain. So it's one of the ways the brain communicates extremely directly with the heart, and that sets up this change in physiology, presumably to allow a physical response to what is seen as a short-term stressor. In fact, we found interestingly that the way that you respond to social stress in the lab, curiously enough, if somebody didn't have really good telomerase, their response was not as ideal as it was if you had a higher level of telomerase. But the heart rate part of it is coming extremely directly and essentially through vagal, through nerve innervation. So it's very direct in that case. The pathways, I hope I've implied, are very complex, from the brain to these quantifiable measures that we have, and we don't want to, for a moment, underestimate the complexity. And the real question is, are there things you can add up that say, aha, this is rate limiting? But heart rate goes up because of the nerves going to the heart.
Let's take, you probably can't hear me because this mic's off. One in the middle here towards the front, and then the one on the row towards the front, and get your hands up quick after those two questions. Please say who you are.
Hi, my name's Stephen Curry. I'm a structural biologist here at Imperial College. I'm concerned about the telomere lengths of staff and students at the college. One of the striking correlations you showed was telomere length versus educational attainment. Would you care to speculate on why there is that correlation?
The explanations have fallen into two classes. One is that education sets you up so that, whatever life is throwing you for decades and decades later, somehow you're better equipped to deal with it. There's one school of thought that thinks in that way. Another school of thought is, what is it that the stark parameter of didn't finish secondary education? Well, what is that? What's the inputs into that? There's a huge number of potential inputs, not just stupidity. That's probably a very rare reason. It's probably much more family, social reasons why someone didn't go through secondary school. So that kind of input starts to be, what is the roots behind that stark fact of not finishing high school? Maybe that starts to be informative, especially since we've started to see in the more extreme cases of cumulative effects of childhood traumas. Now, these are very severe, but we know these effects can be happening and read out as adult telomere lengths. So maybe factors that go into whether somebody doesn't finish high school, secondary school that is, maybe factors that are perhaps milder, but which you can discern when you have these large population studies, maybe those are additively or even synergistically going into that fact. So as I say, two kinds of models. Things were going wrong in childhood that was read out by not finishing secondary school, or an education has you much better able to cope with life's vicissitudes and so on. As we said, it was not related just to them having better jobs, having better income, having better health readouts of various kinds. So it's a fascinating question. That's why the math is so nice, because you just do it as a quantitative thing. And then the theories have to follow the facts.
Hello, I'm Charlotte Mankira. I've just started a PhD here. So you've talked a lot about this causality between shortening telomeres and aging-related diseases. What I'm wondering is, as far as I'm aware, when the telomeres are shortening, it doesn't ever actually enter in normal cells coding DNA or even subtelomeric regions, and the cap doesn't fall off at any point. So why, when the telomeres are shortening, and how does the telomere signal to the cell to change the transcriptional program?
This is why you have great students at Imperial College. You see, that's the question. Why is, the telomere, when they're too short, they actually have several hundred, maybe, a few thousand even base pairs of DNA. I think it's to do with biophysics rates of how you make cooperative complexes. These complexes are very dynamic. I showed them like little rocks sitting on the DNA. It's not, they come on and off with half-lives that have been measured of seconds of coming on and off. So this whole thing is like a swarm of bees buzzing on and off and on and off with the proteins. It's like a swarm of bees and the proteins are buzzing on and off. And so you say, well, this is a telomere, but it's actually turning over all the time. So the biophysics of that, of when it switches into an uncapped state, has to take into account this, and it's a very poorly understood question. We have a terrific parts list for telomeres. We have certain interesting structural possible correlates, but really this sort of two-state almost situation, and it might be some intermediate states, of course, as well, some evidence for that. But the biophysics of that is not well understood, and I suspect it's got a lot to do with how very dynamic, complex things can be maintained by dynamic processes, and then at some point they, cooperatively go into a different state. That's my mental framework.
Yeah,
I hope you can solve this question for us.
There's another research project. We've got time for a couple more questions. Are there any hands at the back there that I haven't been able? Oh, there's all sorts of people waving now. Let's take, there's two at the back there, 2/3 of the way up on the row. Any other hands just to... Okay, after you we will take in the middle, halfway up, and that will probably have to be the end of it. But please go ahead.
Hi, Jess Buxton. What do you think is the reason for the dramatic gender difference in telomere length? And do you think it's to do with hormones, as you showed postmenopausally, you see that big difference?
Yes.
Or do you think there are other factors, such as women having 2 X chromosomes and genes that might be there, or anything else?
We don't know. But exactly as you pointed out, you see, the telomerase gene has some estrogen receptor binding sites in its promoter. So everyone thought, great, it's estrogen. But a lot of estrogen is gone. Now, so whether it's the tiny amount of sex hormones that are left post-menopausally or something else. But this was what was, this is why I love data, right? Because we just looked at it and we said, well, that's what we see with the 100,000 people. So we have to think about it. So we could perhaps be guided now by diving into the genetic variability in the people and looking. So we're hoping that by looking at the data, this will start getting clear. And all of this data is being deposited in NIH's what's called DB Gap. It's a great big database repository. So the idea is it's sort of a resource for everybody to start mining into. And it's very complicated to make this database, because you can imagine all the clinical information longitudinally is very complicated to codify and put into these databases. So it's taking a while, but that's what's going to happen. So hopefully we might get some insights into that question. It was a real surprise.
We're going to take one more. We could go on for a long, long time. It's a pity we can't. But there's somebody in the middle, halfway up. If we could get a microphone up there, I can still see the hand. Can we get a microphone halfway up in the middle? See, exercise is good for telomere.
Yes.
I don't think your mic's working.
So I can speak up?
Yes.
If you stand up and shout, it will be great. Just stand up and shout. Oh.
So if you're making a measurement of the telomere shortening and relating that to different genetic and non-genetic factors in the whole organism, how do you know what type of cell it's appropriate to make the measurement in?
Yes, it's a terrific question. So blood was simply a sample of convenience. We actually did the 100,000 on saliva, with great trepidation. So we were very relieved that we could see things that replicated the previous studies and showed new things. There's certain studies that have related telomere length in buccal cells, that's the inside of your cheek cells, to blood cell telomeres. There's good correlation within an individual, also with skin fibroblasts and overall with blood cells. Now, that said, if we divide the blood cells up, and we've done that into B cells, subclasses of T cells, including senescent T cells, what we can see now is that in an individual, there's a sort of hierarchy, but they move up and down as a group as you go from individual to individual. So that tells us there's two things. There's cell type specific, but there also seems to be systemic control. So as I said, this is a very complex situation. So to me, the miracle is that you can just take this average and start extracting useful information, what I think is useful information. But of course, the diving into the details will be very important for specific aspects of the pathogenesis that may be associated with these kinds of compromised telomere situations. So that's the bottom line. It is cell type specific, but there seem to be clear systemic effects as well. And you can think, well, cortisol will course through your body. It'll, have a lot of cell types it might impact on all at once. That's a simple, oversimplified example. So it's a good question, and as a molecular biologist, I would have said, no hope. You couldn't possibly look at blood cells. They're too much of a mixture. Interestingly, you do see these patterns.
Elizabeth, thank you for those wonderfully clear answers, and thank you for such concise and good questions. I'm now going to ask Professor Maggie Dowman, the Dean of Faculty of Natural Sciences, to express on our behalf her thanks.